These are the materials that you need for this lab. First, the hot plate, tape, stirring rod, multimeter, candle, alligator clips and glass slides, matches, tweezers, petri dish, and the Q-tips and the berry juice, which will be provided by your instructor. To start off with the experiment, you have to figure out the conducting side, which is the rough side. You will know it's a conducting side by using a multimeter and setting it to 200 with a Greek symbol below. The conducting side will have a resistance of 20 to 30 ohms. With the conducting side up, tape the glass on 3 sides. Wipe off any fingerprints or oils using a Q-tip wet with ethanol. Add a small amount of titanium dioxide paste, around 2 to 3 drops, and quickly but carefully spread by pushing down and across with a stirring rod before the paste dries. Allow the slide to dry for 5 minutes before carefully removing the tape. Make sure it doesn't scratch the titanium dioxide coating. Place the slide on the hot plate with a pair of tweezers and heat the slide at 450 degrees Celsius for 30 minutes. Make sure the slides turn yellow and then white again. Allow the glass to cool by turning off the hot plate and don't place on the table for 10 minutes to avoid cracking the slide. Get a match and carefully light it on fire and light a candle. Choose one that has a long stick in order for it to last longer. Make sure to immediately blow it out. For any discrepancies, call your instructor. Get another slide with the conducting side down and pass it through the candle flame with the conducting side being coated with carbon. Lastly, wipe off carbon along the perimeter of 3 sides of the carbon-coated glass using a dry cotton swab. Place both slides side by side into a petri dish and leave for tomorrow if there is not enough class time.
Place your blackberry juice in a petri dish. Place your titanium dioxide glass slide onto the blackberry juice. Be careful not to push it in, just lightly place it on. Rinse the slide gently with water to remove any berry solids and then with ethanol to remove the water. The ethanol should have evaporated before the cell is assembled. Assemble the 2 coated glass slides together, but it's extremely important to make sure there's space for the alligator clips to be placed on one piece of glass on each side. Do not rub or slide the plates. Clamp the plates together with binder clips. Add a drop of triiodide solution to opposite edges of the plate. Then, wipe off excess solution from the exposed areas of the glass using Q-tips dampened with ethanol. Important: the cell will not work if there is any solution left on the exposed areas where the alligator clips attach. Set the multimeter to 2000 millivolts to measure the voltage produced by your cell. Attach alligator clips to the multimeter wires and the cell. Place your cell in the light to find the voltage. If you notice that your voltage is negative, switch your multimeter wires and find the voltage again.